Right, so hello everyone, and today I would like to tell you a bit about how can genome mining from space-dwelling bacteria help us to stay healthy while colonizing other planets, and that's from bioinformatics perspective. So, as we are approaching 7th decade of space exploration since we first launched as a Sputnik satellite to the orbit, we quickly realized sometime around when the Apollo program was starting that apart from looking for signs of extraterrestrial life, we should be also concerned about bringing terrestrial life to space. Especially now, since we are planning to set foot again on Moon and go to Mars and beyond. And this is why, because some of the very early during the space program, it was found out that there are some microorganisms, especially, that are capable of surviving extreme conditions, and are thus it is possible to for them to be in space. So one of these first organisms was Bacterium Deinococcus radiodurans, which was actually found after the food preparation for astronauts in the early days, and that bacterium was found to survive the ionizing radiation that was treatment of this food. Now, as much as having an alien life form is that could pose a threat to us is very unlikely and still remains in the science fiction realm, the other problem might be those bacteria. So the Deinococcus radiodurans is actually quite neutral. It's not harmful to us. But what if there is another bug that would be hitchhiking with us and can cause us problems like disease outbreaks, or if we, especially thinking about like long-term space voyages, could succumb to something like Martian scurvy, right? So that is actually something worth considering, and this is especially now becoming apparent since we are planning these long-term space missions. So one of the programs that space agencies are conducting is this microbial tracking mission, because International Space Station, since its launch, has been frequently populated by crews that were changing different time points, and obviously a lot of microorganisms have been brought together with them, and they do grow on various surfaces of the ISS. So microbial tracking missions are especially interested in those biofilms covering different surfaces of ISS, and those samples have been taken from the ISS, brought back to Earth, and in the biological labs, their microorganisms have been isolated, sequenced for the taxonomy identification to find out what those species are and to characterize them functionally. And one of those studies is the one that I was participating in, and we were given samples of genomes of 5 different species that were collected from these biofilms from different locations of ISS, so crew quarters and arete, which is a fancy name for toilet facilities. These microbes are different species. 4 of them are non-spore formers. One is known to form spores. They have different genome sizes, different amount of genes, and we managed to characterize almost all of the genes in all of the organisms using bioinformatic tools such as DeepFry, which helped us to understand what functions do they have. So I will come back to one of those species in the later slide, Arthrobacter burditzki, because of those characteristics. So we found that there is a pool of novel genes that those microbes have in common, even though they are not so closely related to each other. And there are some among the functions that we try to characterize. There are many signal transduction, replication, translation related functions, but also novel metabolic functions. And novel metabolic functions have been discovered in the form of biosynthetic gene clusters in the genomes of all of the species that we studied. And in the case of Arthrobacter, this commonly looking bacterium that I showed you before, we found the evidence of novel antibiotic family that is quite interesting. And apart from that, there is a multitude of different metabolites, secondary metabolites, carotenoids, metallophores, terpenes that we could potentially investigate in the context of mitigating health risks and also supplementing astronauts in the space voyages. So this is where the idea of AstroFarms was born, where we try to develop the optimal food to nourish astronauts and protect them from harmful space environment. So we started doing that together with my co-founders, partners, looking at the microbial consortium coming from kombucha. So symbiotic community of bacteria and yeast have been selected based on the properties that they may prove to be the most beneficial, and we are now trying to extract bioactive compounds from those communities that are able to survive simulated space conditions, so increased radiation and also presence of regolith minerals, so the moon or Martian rocks. And we do that by now isolating those microbial single organisms from those microbial communities, characterizing them, and also trying to find out what are the key bioactive compounds that those guys are synthesizing, and then trying to design in vivo assays of pre-selected compounds on cell cultures, and then pursue the metabolic studies on healthy volunteers to obtain a library of compounds with potential health benefits that can be applied both for space missions, but also on here on Earth. And to summarize my talk, I would like to say that microbes isolated from space are those, although they are coming from Earth, they are novel species, but they have different similarity to terrestrial organisms. And the genomes contain genes encoding novel proteins, which we were able to identify thanks to bioinformatic tools. And those novel metabolic pathways are of particular interest because they contain a multitude of compounds with potential health benefits. And their analysis may help to identify genes for survival under extraterrestrial conditions, but also in case of encountering some potential pathogens, we can also target them using those genes that we know are essential for them to provide this survival. So with this, I would like to thank you for your attention.